an electron microscopist, and I'm interested both in inorganic nanomaterials as well as biological nanomachines and understanding how they work using projective single particle electron microscopy. And most of these techniques are projective, at least in the biological sciences, because it's transmission electron microscope, so the electron wave traverses the specimen and we get the projection image. Ernst Ruska invented the electron microscope, and his construction is very similar in principle to the electron microscopes that we are using today. We have a condenser lens system that is focusing the electrons onto the specimen, and then we have an objective lens system that is creating a magnified image of the specimen. And in Ruska's times, you collected your images on image plates. Today we have very bright and coherent electrons from field emission guns. It's the brightest electron source in the known universe. We have extremely stable stages because we're doing atomic resolution imaging, so we can't move around too much. And over the last couple of years, we have seen a revolution in detector technology with these CMOS-based detectors that can record movies and these movies have better digital quantum efficiency than the traditionally used film. And so we freeze the molecules in vitreous ice by plunge freezing them rapidly into liquid ethane. So all the molecules are randomly oriented in this ice layer. And so these are very electron beam sensitive specimens, so we can only shoot them once. And so we don't know from which direction we are looking at these molecules. And so the grand challenge is, of course, in the biological sciences, we are purifying macromolecules. We have a large amount of them. We assume them all to be identical. We image them, and then we get these little projection images here, and then we have to figure out, it's an inverse problem, obviously, how they are oriented with respect to each other in 3D space. And if we are doing that really successfully, we can reconstruct atomic resolution densities, or at least densities that allow us, based on the knowledge that we have of protein molecules, to figure out the atomic distribution. Another problem that I got interested in a while back is, what do we do when we have molecules that we cannot purify to homogeneity? This could be, for example, metallic nanoparticles. When you synthesize them, every molecule is unique. And so we try to develop a method where we can enclose these nanoparticles in liquid, and then we shoot electrons onto them, and they spin around in solution. And I'll show you in a while that we can capture different views of the nanoparticles and then reconstruct in 3D from a series obtained from a single molecule. And just to give you an idea about the challenges, this is cryo images, typical cryo, probably the best cryo images that we can obtain today of the TRPV1 membrane receptor complex. And I would estimate that the signal, the noise variance is probably 10 to 20 times the signal variance. And one problem that we usually solve first, at least in biology, is to try to solve the simpler problem of grouping all of these differently projected views into clusters in order to be able to average over the cluster centers and increase the signal-to-noise ratio. And so these are ribosome projection images here. And of course, we don't know from which direction we are looking at them, but we know that it's oversampled because we have a large data set of many copies. So it's meaningful to cluster them. Their rotational origin will be shifted in the box, and they will be arbitrarily rotated in the plane. So we have 4n parameters, where n is the number of particles that we need to determine in an unsupervised manner. And this is a quite difficult task because we need to deal with the microscope aberrations, and we need to deal with the high level of noise. And this is sort of the traditional approach that people have been taking, assuming that you have a guess of your 3D density. You can project this density into known orientations in 2D and match them to electron microscopic projection images. And so the challenge is then, if we don't know or we don't have a good guess, here I just reconstructed from random orientations the density. How can we design an algorithm that would be able to identify the orientations when we don't know the answer? And so I suspect it would be some kind of an iterative approach, and we have developed such methods, and I'll just mention them briefly in the next slide. So the standard method then is deterministic in the sense that it tries to identify for every image which is the best orientation, and it assigns one hard orientation. And of course this is a bad idea when the model is of very poor quality and the data is very noisy. So what we are doing is we try to assign a distribution of orientations to every particle image and then weight them in the Fourier domain. And then we use stochastic optimization to find a good weight distribution. And if we succeed with that, we can reconstruct biological macromolecules to very high resolution. Here you see the proteasome and then the polypeptide backbone fitted into the density map. And I've been working a long time with these problems, but then I have some friends in material science that synthesize nanocrystals and managed to enclose solutions of these nanocrystals in a bag, one molecule thick. So it's a graphene bilayer where we put a solution of nanocrystals. And the idea was can we just direct the electron beam onto these individual nanocrystals, and then since we have this wonderful detector technology now with down to 1 millisecond time resolution, perhaps we would be able to capture different views of these nanoparticles as they are spinning around in solution. And it turned out to be fairly difficult because graphene is hydrophobic, so it tends to produce a very round droplet, so it's not electron transparent because it's too thick. But around the edges we managed to find some areas where we could collect images, and this is what it looks like. So here you see all the individual nanocrystals, and you see they are vividly heterogeneous in different sizes and so forth. And these big ones tend to dwell sort of along a single zone axis, whereas the smaller ones are rotating very rapidly. I'll just focus in. You see the big ones tend to stay pretty much still, whereas the small ones are spinning around. And just to give you a better idea, I just slowed down the process where you see this nanocrystals displaying different views to the electron beam. And so when I saw this, I called it beam-induced tomography, because I think that's exactly what it is. And we managed to figure out, I mean, there's obviously lots of room for improvement. This is very challenging problems. But we managed to reconstruct 3D density maps so we could propose tentative atomic positions. And what we saw was that these nanoparticles have multi-domain structures, and this is because of the way that they are formed. They are formed by multiple coalition events. And something that we're interested in doing as well, because we have time series data from these detectors, and now the time resolution has become even better, so we could potentially look at the structural changes that these particles undergo as they interact with the electron beam. And in our preliminary results indicate that there is a big deposition of energy into the graphene liquid cell initially, which causes a disorder of the particles. And then they sort of start to interact with the electron beam and re-anneal into a more ordered state towards the end of the series. But I mean, here you could envision more sophisticated sorting algorithms where you would sort of trace out individual atomic movements throughout the series. And then back to the biomolecules, which initially caught my interest. And the assumption that this 3D reconstruction methodology relies on is that these are nearly identical copies. This is unfortunately seldom the case. We refer to heterogeneity as not all images representing projections of the same object. And it's very common to have compositional heterogeneity. For example, you have a factor binding to a larger complex and dissociating. You can have random loss of components, which are artifacts of purification and specimen preparation. We have to account for these variations if we want to have high quality density maps. Protein molecules also move to exert their functions and often undergo machine-like motions, characterized by swinging arms, rotating domains, and breathing motions. And we have tried a large number of different algorithms for dealing with this sort of stuff, but I'll just speak briefly about one. So what we did here was we took 4 different molecules. We mixed them in a test tube and we collected the data just to validate what we were doing. And we could apply the 2D clustering that I talked to you before about in order to resolve the different states in 2 dimensions. But of course, a 2-dimensional algorithm could never distinguish between different structural states. So these belong together, right? This is an IP3, it's the receptor we have in our muscles that transports calcium to create gradients. These 2 belong together, And here we have a ribosome, ribosome, ribosome. So how could we automatically figure this out? Well, we would have to have a three-dimensional metric. And so we turned to the Fourier slice theorem, which was discovered by an Australian astronomer in 1956, Bracewell. And it states that the Fourier transform of a 2D projection is a plane through the origin of the 3D object Fourier transform. And that means that any two non-parallel projection directions, they share a common line in Fourier space. So we went ahead and developed a two-pass clustering procedure for sorting states in 2D, whereby we, for every pair of images, we fix one in an arbitrary orientation, and then we find the orientation of the second one, which gives maximal correlation, and then we pick the maximum and we use that as a metric of similarity between a pair, and then we just create a matrix that we subject to clustering, and out comes the different states, and then we can use standard reconstruction techniques to reconstruct the different piles, and and we tried to mix these molecules in different ratios also just to test out. Really, this was just a proof of concept experiment for some biological problems that we are struggling with. For example, would it be possible to, instead of investing all these efforts into purifying the macromolecules, could we just take lysate from a cell, or could we take blood and fractionate it and look at that in the electron microscope and sort out the different states? I mean, we are not there yet, but perhaps with your help we will get there. So thank you for your attention, and also thanks to my lab at the BDI for all their help.